Adrenocortical cancer is a rare cancer, and right now we only have really one standard treatment, which is a combination of chemotherapy and a drug called mitaine. And there is a need for more treatment in this setting. And in Vancouver, we have been doing a much larger initiative where we sequence the genome and the transcriptome, that's the product of those genes, from patients with advanced cancers. And so as part of that, we sequenced 6 patients with adrenocortical cancer. So there are a number of studies out there that have used different methods for sequencing. Our approach is to do whole genome and transcriptome. And so rather than having a panel-based approach where you ask specific questions, we basically sequence all the DNA in a sample and all the RNA. So this is a small population, and we have found a number of things that have been described by other groups. There's sort of known mutations in these cancers, and we have found a number of these. We've also found some mutations in pathways that affect the cell cycle and some mutations in DNA damage repair that may suggest different treatments. Well, I think it's a small piece of information that has to be put in context of sort of the larger literature out there. And we're still hopefully accumulating more and more information as we get more patients and as we get more data. Eventually, this can lead to clinical trials that can be guided not just by the type of cancer, but by what the genomic findings are. I think we're still at a stage where we're trying to describe the landscape of these cancers, though. I would say that we're now just starting to understand that these cancers are very different from patient to patient, and we have one cohort. There are other cohorts out there that where the methods are a little bit different, but essentially with the same overall aim of describing what are the genes driving those cancers. And I think ultimately we're now understanding that there are a few sort of key things that happen in a number of patients, but patients by and large, from patient to patient, are very different. And so that's why it's very challenging to do studies where all patients may be getting the same treatment when it may only be the appropriate treatment for a subset of those patients. So we found some alterations in cell cycle pathways and in DNA damage repair that had not been previously described in the literature.